I'll give it one more minute. Okay, I think we're gonna go ahead and get started. Can people hear me okay? Great. So on behalf of Alexandra Newton, who's the director of Cell Signaling San Diego, I'd like to welcome everybody to the Roger Tsien Cell Signaling Distinguished Lecture Series. So this lecture series was created by Center of Cell Signaling San Diego to honor the legacy of late Roger Tsien. Roger was a true pioneer. He created and developed a series of ingenious molecular tools that revolutionized neurobiology and cell biology. With his forward-looking vision, profound knowledge, and unparalleled creativity, he laid out the foundation and the conceptual framework and impacted his thinking for many fields, including cell signaling field. He has such profound influence on the scientific community and many of us personally. So this lecture series was created to honor the legacy of Roger Tsien. And building on Roger's legacy has been one of the driving forces for establishing Center of Cell Signaling San Diego. This new center unites researchers who have a passion for mechanistic understanding of cell signaling, and with a goal to leverage that understanding to treat disease. And today's lecture exemplifies how impactful that mechanistic understanding of cell signaling could be. And for that, we're grateful to Bristol Myers Squibb for sponsoring this lecture series, and we look forward to collaborating for many years to build this lecture series into one of the pillars of our community. Now I'd like to turn the podium to Susan, Distinguished Professor of Chemistry and Biochemistry and Pharmacology, to introduce our today's lecturer, Professor Dario Alessi.
So it's a real pleasure to have Dario here today. Like Roger, Dario is a real pioneer, and I think he began actually at the University of Birmingham, I think, synthesizing spin-labeled nucleotides, and then went to, as a postdoctoral fellow, to Philip Cohen's lab in Dundee, and that sort of changed the course of his life and influenced all of us. And he's the ultimate biochemist, and I think one of his big discoveries there was a really huge challenge to find out what was phosphorylating AKT. And from massive purifications of huge amounts of tissue, he identified PDK1 as the activating kinase. And then another really pioneering experiment that he did then was to identify the site on AKT that was phosphorylated with using tons of P32, which we did back in those days. And so he's really undaunted by challenges. And I think he began his work, well, then he also showed that LKB1, the tumor suppressor kinase, is the master kinase that activates AMPK. And so these are all major steps that have really advanced the signaling community. And then in 2005, he began working on LRRK2, which he says is probably the most challenging of all of the projects that he's undertaken. And it took like 10 or 11 years before he really started to identify what are the substrates that LRRK2 phosphorylates. And so we're going to hear the latest from his work on LRRK2 and Parkinson's disease, and it's just such an honor to have you here.
Well, thank you very much. Well, okay, well, thank you very much, Susan, and to Alexandra for the incredibly kind invitation to deliver this lecture. I personally never met Roger Tsien, but I heard him talk at several conferences in Europe, where he sort of dazzled me and the rest of the audience on all the technologies he was developing to investigate biology. And I always remember him describing the rainbow of different colored GFPs, and that slide just completely blew my mind and inspired me so much. And I know how much he means to all of you here, and how dear he is to all of you, and how much you miss him. And so it's a great honor for me to deliver this second lecture. And I just want to, before I go into my science, to say, for me, whose interest is understanding cell signaling pathways and how they're controlled and how they regulate biology and disrupted in human disease. When I look at what's going on in the world in this field, without a doubt, what you have in San Diego is the greatest constellation of researchers who work in this area anywhere in the world. And but maybe you don't know it, but it's a superb talents you have of people working on cell signaling pathways. And I'm really pleased to hear that you're organizing yourselves into cell signaling community and infrastructure, and that's going to reap, you'll get great rewards from this. So in today's talk, I'm going to tell you about the work that we've been doing on this LRRK2 protein kinase. I should also say that San Diego is probably the place in the world where there's most research going on in LRRK2. I think I count at least 6 groups that I know of here working on LRRK2. There's probably more. And I think I'll acknowledge a lot of the work as I go through my talk. But the reason we became interested in LRRK2 is because it was a sort of unstudied kinase in 2005, which was strongly linked genetically to inherited Parkinson's disease, and dominance mutations either in the kinase domain or the adjacent GTPase domain were clearly linked to causing Parkinson's disease. And it's it's got a very interesting architecture. As you can see, it's one of the only kinases with LRRK1 and a few other kinases that actually have an unusual GTPase domain located next to the kinase domain that's known as a rock core GTPase domain. It's actually got 3 subdomains that you'll see come together to form a GTPase domain. And the Parkinson's causing mutations, at least at the time, were either in the GTPase domain or the kinase domain. And it's now known that all the mutations lead to activation of the kinase. So it's clear that too much kinase activity over years of life is leading to Parkinson's disease. And we now, obviously, through the work done in San Diego, know a lot more about the structures of LRRK2. Originally, I think about 2 years ago, Elizabeth Villa described the first and still the only active form of the LRRK2 in the in situ tomography structure of the enzyme, which is an active dimer. And then Andreas Letzen's lab described the RCKW domain. This is the C-terminal catalytic region of the protein, where you can see beautifully the structures of the kinase domain. You see this is the GTP binding domain of the ROC domain, and you have the core B, the core A, the kinase domain, and the WD40 domains. And this structure is the confirmation of the inactive LRRK2. And another lab, Ji Sun's lab, recently published the structure of the inactive form of the of full-length LRRK2, and you can see it has this very long, anchorin, leucine repeat, anchorin domain, and armadillo domain that forms a huge long tail, outside the structure of the enzyme. So, so it goes to Parkinson's disease. I'll tell you one slide about Parkinson's disease. It's it's a very serious disease. It affects nearly 10 million people, in the world. Many people who get it are in the later on in life, 60s, 70s, but it also affects a lot of younger people who are often in the prime of their lives. And, the sad thing is, although you can treat some of the symptoms of Parkinson's disease, there's nothing that slows down the progression of the condition. And I guess the aim is, if you can understand pathways involved in Parkinson's disease, you can maybe play the engineer to develop treatments that counteract the mutations and might start slowing down the progression of the disease, or maybe even in the future, actually stopping Parkinson's disease from forming, if you can tell people who are at high risk at developing the disease. And this is exemplified by the most commonly used drug to treat Parkinson's disease is a compound called levodopa that replaces dopamine therapy, and this was developed in 1967. This is the year I was born, and most other drugs developed since then are sort of incremental improvements to the delivery methods to deliver dopamine therapy. So there's an urgent need for new therapies in this area. And for LRRK2, one of the reasons that it's really important to study, it's one of the most common genetic causes of familial Parkinson's disease. So depending where you live in the world, it's anywhere between 1% and sort of 5% of people with Parkinson's disease have the disease driven by mutations that activate LRRK2, and symptomatically it's very similar to the common sporadic form of the disease. So it affects people, generally speaking, later on in life. It's not an early onset form. So people in their 50s, 60s, 70s, develop the disease. And by far the most common mutation is the G2019S mutation that Susan Taylor showed beautifully was sort of stabilizes the kinase in a more active conformation in which the catalytic spines are all well aligned to activate the kinase, and this affects 95% of people with LRRK2 mutations have this mutation. And there's also data emerging from various laboratories that there's evidence that LRRK2 might also be activated in some patients who have this so-called idiopathic Parkinson's disease, in which it's not known what the cause of the disease is. So when we started working with LRRK2, the key question is, what was the cellular substrate of this enzyme? And this, I find substrates of many kinases. I actually give a lecture to our undergraduate students on how to find substrates for a protein kinase. And this was the hardest. I think it was the hardest because LRRK2 in cells has very, very low activity. And when you did knockouts and you made knock-in mutations, or you used inhibitors and you did mass spectrometry, you had lots and lots of hits, and many of those turned out to be artifacts from different clones, different mice, or off-target effects of the inhibitor. And the way we sorted this out, make a insensitive mutation of LRRK2. So we made a mouse in which was insensitive to a specific inhibitor of LRRK2. And then you give the inhibitor to both the wild type and the drug-insensitive mouse. So you give, and then you only look for a target that goes down in the wild type, but not the drug-resistant mutant. And this was the key. And we worked at this time, I'd burnt all my bridges with lots of mass spectrometers who had helped us before, but we were working with Matthias Mann at the time, and this enabled us to identify Grab10 and Rab8, a related Rab, as the key physiological substrate of LRRK2. And so Rab GTPase is a key regulator of membrane biology. And what's very interesting is that the LRRK2 phosphorylation sites right in the middle of the business end of that controls the biology of Rab proteins. And this is the switch 2 motif that's shown in blue here, and then this is the effector binding motif of Rabs, and the phosphorylation of the Rabs in the middle of the effector binding motif obviously influences their function greatly, and I'll come to that. So yeah, if you look through a cell, as you all know here by EM or method, all you see is membrane after membrane after membrane, vesicles. It's incredible the membrane biology that's within the cell. And this is all orchestrated by a group of about 70 different Rab proteins and the proteins that regulate the Rab proteins. And I think there's still a very little insufficiently studied area of biology, and there's obviously a lot of important things to learn about how Rabs control, membrane biology. So I just want to show you some data just to really persuade you before we move on that Rabs are really the key substrates of LRRK2. And I like this slide because it's a phosphatase gel, and you separate out the dephosphorylated Rabs from the phosphorylated Rab10 in this gel. And this immediately tells you that, yes, indeed, the LRRK2 activity is extremely low in cells. So only about 1 or 2% of the cellular Rab protein is actually phosphorylated by LRRK2. And this is one of the reasons why it was sort of so hard to identify. And when you treat with this specific inhibitor called MLI2, you induce dephosphorylation of Rabs around 3 nanomolar. You've lost the phosphorylation of the Rab proteins. And when you do the same experiment, this is an inhibitor resistant mutation, you can now see you have to go up to 100 nanomolar MLI2 to induce the dephosphorylation of the Rab protein. So this gave us confidence that the Rab proteins were indeed the key substrates. And the other results we found very quickly was that all the 7 pathogenic mutations I showed you in my introductory slide, when you made knock-in mice or you obtained knock-in mice with these mutations and tested Rab protein phosphorylation in cells or tissues derived from these mice, you see an elevation of Rab protein phosphorylation. This is what you would expect if Rabs are the sort of genuine physiological substrate. And obviously, if you treat with an LRRK2 inhibitor, such as in this case, this was a GSK compound, you reduce the Rab phosphorylation to nothing in both the wild type as well as the pathogenic knock-in mutation. We obviously now have much better technologies to measure some of these Rabs. We worked with the Michael J. Fox Foundation and ABCAM to make a series of monoclonal antibodies that are extremely useful and widely used to study Rab10 phosphorylation, Rab12 phosphorylation. And we also found that, that there's about 50 of the Rab proteins out of the 70 that are found in the human genome. About 50 of them have this conserved serine or threonine residue in the middle of the switch 2 motif. So we tested all 50 of them to see which ones would be phosphorylated by LRRK2 in overexpression systems in cells. And the hits, we then tried to look at the endogenous protein as well to see if it was phosphorylated by LRRK2. And the group of about 12 or 13 Rab proteins I have in this slide, we're fairly confident that the endogenous protein is phosphorylated at the equivalent site to which Rab10 is phosphorylated, and this is modulated by LRRK2 inhibitors and activating mutations. And we now also have recently developed, I won't go into this assay, a mass spectrometry PRM assay that enables one to quantitatively analyze the phosphorylation of the group of Rab proteins that are shown in red here using mass spectrometry, which is complementary to the phosphoantibody approach. But obviously it's more quantitative and more laborious, but you can analyze the phosphorylation of most of the Rab proteins that are phosphorylated by LRRK2 in a cellular or tissue experiment. So I just want to highlight some recent work that we've done on the variants of LRRK2 that might be implicated in human disease. And with some genetic experts, and we assembled a list of about 98 variants that are shown in this slide that are found in not only just the GTPase domain and the kinase domain, but that are found in all the domains of LRRK2. And for a lot of these variants, the genetic evidence that these are linked to Parkinson's disease is weak and not very strong. So what we did is we simply made all these variants and tested how this impacted the catalytic activity of LRRK2. And this has resulted in us finding another 23 variants that are shown on this slide that we're quite confident also directly stimulate LRRK2. So there's a lot more variants compared to the original list. And instantly, some are located in the armadillo ankyrin domain, the leucine-rich repeat domain. None are located in the core A domain. And there's one clear-cut variant that's well studied in the WD40 domain. And below is this slide that shows that the all the variants that are listed here sort of stimulate the Rab10 phosphorylation compared to the activity that one sees in the wild type protein. So I think this sort of information might be interesting with the structural biology that's going on to try and understand how these variants, especially those located out with the ROC and the kinase domain, might influence the function and the structure and then the function of LRRK2. And as you can see, most of the variants in the GTPase domain locate either in the ROC domain or the core B domain that is highlighted here. And the structural work of Andres' group and also Ji-Sun's group really reveals that these mutations all lie within the interface between the kinase, between the ROC domain and the core B domain. And all these mutations seem to be destabilizing this interaction. So this suggests that destabilizing this interface is somehow leading the LRRK2 into a conformation that can maybe better adopt the active conformation, by promoting the fully active conformation. And there's only one exception to this to this interface, and this is mutations of arginine 1728, which here these mutations are located between the core B, core B dimer that's seen in Elizabeth Willer structure, and these this residue is mutated either to a leucine or to a histidine residue, and this is quite a low resolution structure, so I don't think it's really clear to understand how this mutation impacts the activity, but this one also potently activates LRRK2, even in the recombinant protein. When you immunoprecipitate the protein with this mutation, you see a very robust activation of the of the kinase. So we're going to move on a little bit now, and the next question was, okay, the Rabs are phosphorylated, but what is the physiological role of this phosphorylation? And as only 1% of the Rab protein is phosphorylated by LRRK2, it is pretty clear to me that the way this was going to work would be that the phosphorylated Rabs would be able to bind to a new set of effectors that the dephosphorylated form doesn't bind to. And this explains why, let's say, 1% of the protein would then be able to have a new dominant function. And if it was the other way around, it was inhibiting the interaction, the cell, I don't think, would care if 1% of the phosphorylated protein was phosphorylated to 3%. So because of this, we searched for proteins that would bind to the phosphorylated Rab proteins. And what we found, and I think there's going to be a lot more of these proteins, but so far what we found is that there's at least 4 proteins that I've shown here that the phosphoRab8 and phosphoRab10 bind to. They all have these are all identified biochemically from mass spec pull down experiments, and they all have an RH2 domain. And this is a sort of it forms a dimeric alpha helical region, and this is the binding site for the phosphorab proteins. And we've done some mass spectrometry, not some structural analysis with Amir Khan's group in Trinity College in Dublin, where we made peptides that correspond to these RH2 domains and co-crystallized them with phosphoRab8. And you can see how the phosphoRab8 binds to the dimer of the alpha helix. And there's some basic residues towards the top of this alpha helix region that form critical interactions with the phosphoRab8 protein. And the orientation of this complex is also nice, because the Rabs would be binding to membranes and oriented in this direction, and this lines up very well with the orientation of the RH2 domain, sort of pointing away from the from the membrane. So and my collaborator in Stanford has done a lot of work. Susan Pfeffer has done a lot of work on this, on understanding the role that the RILPEL1 phosphoRab10 complex plays. And the RILPEL1 was previously known to be controlling ciliogenesis. And it turns out that this pathway probably does control cellulogenesis, and at least in cholinergic neurons in the brain. And when you get this complex of phosphoRab8 or phosphoRab10 bound to RILPEL1, this results in inhibition of cellulogenesis. And we got very good evidence for this. And you can use this, so you use LRRK2 inhibitors, you can show that you can promote cellulogenesis. And if you inhibit cellulogenesis in neuronal cells, you get reduced response to hedgehog signaling pathways that can also then feed back to the dopaminergic neuron cells that produce the dopamine and survival pathways. So this could be one possible mechanism by which LRRK2 could potentially be linked to Parkinson's disease in the brain. And this work's all been driven by Susan Pfeffer. So yeah, the model of how this downstream pathway works that I think still needs some more work is that, the Rab proteins at the membrane, when they're in the GTP bind form, they'll bind to effectors. And then LRRK2 comes along. And yeah, I probably didn't say this, but once the Rab proteins are phosphorylated by LRRK2, since the phosphorylation site's in the middle of the effector binding domain, the effector can no longer bind to the Rabs because that binding is disrupted by the phosphorylation from the Rab protein. And then we can see this experimentally. All the effectors we've tested that bind to the dephosphorylated Rabs no longer bind to the phosphorylated Rabs. And this includes various Rabin effectors, as well as the GDI, which is the chaperone protein that transports Rabs between membrane compartments. And then once the Rab protein is phosphorylated, it can bind to a new set of effectors, some of which have been identified, most probably haven't been identified. And then this will trigger different biologies that would be interesting to identify. And then probably this complex of the Rab bound to the effector proteins will then bind to a new group of proteins. Some might be involved in cellulogenesis, some might be involved in lysosomal or mitochondrial biology that we're trying to uncover using mass spectrometry approaches at the moment. So coming to what's upstream of LRRK2, is also an interesting question, is how is LRRK2 activated? And obviously using Rab protein phosphorylation, this can be used as a monitor for the activity state of LRRK2. So what we know here is that if you put LRRK2 at the plasma membranes of cells, you target to the Golgi, lysosome, mitochondria, plasma membrane, it seems to become activated. And you can see this by increased autophosphorylation of LRRK2 at a site called serine 1292. And you also promote Rab protein phosphorylation because you bring LRRK2 in close proximity to the Rabs, where they're localized at the membrane of cells. So this is going to be really critical. People in the field think that movement of LRRK2 to a membrane is the key step in controlling the activation of LRRK2. And I think most investigators agree that most of the LRRK2 is cytosolic, and only about less than 10% of the Rab of the LRRK2 at any one time is located at the membrane of the cell. So our working model at the moment is that LRRK2 is recruited to membrane vesicles or various membranes by a recruiter Rab that binds to the armadillo domain. And this brings LRRK2 to the membrane of cells, where this brings it in proximity to to its Rab substrates. And this probably also activates LRRK2 by some unknown mechanism. It could be dimerization, it could be binding to another partner, because you see increased autophosphorylation of the Rab protein, of LRRK2 when it's recruited to the membrane of cells. So the first model where we found this mechanism was looking at a protein called Rab29. This is also implicated genetically in Parkinson's disease. And what we found is when you overexpress Rab29 in cells, you got a very big activation of LRRK2 and enhancement of Rab protein phosphorylation. And it turns out that Rab29 binds to the armadillo domain of LRRK2 and recruits it to the trans-Golgi network. And this results in increased autophosphorylation at serine 1292 and enhanced Rab10 phosphorylation. And we mapped the Rab29 binding site to a small region in the armadillo domain between residues 350 and 550 as the as the key binding site. And we've tried quite hard to try and crystallize Rab29 with this fragment, but we didn't succeed. So what we've done since AlphaFold has come out, we've taken this fragment of LRRK2 and used AlphaFold to predict what the binding surface might be. And obviously we get a very persuasive model coming from this analysis. And the good thing about this model is it predicts some clear-cut residues that might mediate this interaction. And then you can test this hypothesis by mutating these residues in full-length LRRK2 and seeing whether this affects Rab29 binding. And in the cellular localization experiments, this is what happens. So the wild-type LRRK2, when you co-express it with Rab29, goes completely to the Golgi. 100% of the LRRK2 binds to Rab29, which is located at the Golgi. But when you introduce many of these mutations that I showed you on the previous slide, you completely block the localization of LRRK2 to Rab29. And when you do the activity-based assays, you also see a very similar result. So the wild-type Rab29 stimulates LRRK2 activity, so you see enhanced Rab10 phosphorylation. But when you mutate these residues, these 4 residues that are predicted to mediate Rab29 binding, you largely lose the activation of LRRK2 by Rab29. Interestingly, for some of these mutants, you also seem to decrease the basal activity of LRRK2 without the Rab29. And we have some evidence, and Susan Pfeffer is also doing a lot of work on this, is that that this binding site for Rab29 also binds to other Rab proteins like Rab8 and Rab10. And we think that this is a general binding site to recruit LRRK2 to vesicles or membranes of cells, and bringing LRRK2 in close proximity to where its physiological substrates will be. And we call this the dephosphoRab binding site. And Susan Pfeffer has been doing tremendous work that will hopefully be published soon, and she's discovered that there's a second binding site at the N-terminus of LRRK2 that binds phosphoRab proteins. And this will be important probably in the feed-forward mechanism that once you get some of the LRRK2 that's phosphorylated, some Rab proteins that are phosphorylated the membrane of cells, you recruit more Rabs to the phosphorylated, Rab proteins, and you'll stimulate, recruitment of more LRRK2 to the membranes where you can phosphorylate more Rab proteins in a feed-forward pathway. So the other activating mechanism that we found that still requires further work is we did a screen to test whether any other mutations in genes that cause Parkinson's disease could activate LRRK2. And the top hit from this screen was a component called VPS35. So, VPS35, this is a component of a complex known as the retromer complex that is composed of a pseudophosphatase called VPS29 and a cargo binding protein called VPS26. And the key role of this complex is to regulate retrograde trafficking, moving proteins, from the endosomes to the Golgi, apparatus. And interestingly, there's an aspartic acid, which is actually quite far away from the cargo binding domain called aspartic acid 620. And when this is mutated to an asparagine, people get Parkinson's disease. In fact, it seems to be more severe than LRRK2 because the people get the disease on average 10 years earlier. And how this mutation causes Parkinson's disease is really unclear. So we got hold of a knock-in mouse that carries this mutation. And when we look in the cells or the tissues of the mouse, we saw quite massive increase in Rab protein phosphorylation. In fact, this is much more than a bigger increase in Rab protein phosphorylation than you see with any pathogenic mutation in LRRK2. And this increased Rab protein phosphorylation is also blocked with an LRRK2 inhibitor. And we also see this in patients. You can actually isolate monocytes from people who have mutations in VPS35, and then you can measure Rab10 phosphorylation in the monocytes from the people with these mutations. And we like monocytes or neutrophils is because these cells have high levels of LRRK2, and you can isolate very homogeneous cells from a patient. So it makes the analysis much better than if you're using PBMCs. That's a sort of a heterogeneous group of cells. And doing this, you can see that the 3 subjects who have the VPS35 mutation have enhanced Rab10 phosphorylation in monocytes. That's brought down to basal levels with an LRRK2 inhibitors. And if we compare them with age-matched people with idiopathic disease who don't have mutations in LRRK2, they have sort of normal low levels of of Rab protein phosphorylation. And so more work is needed to understand how VPS35 stimulates LRRK2 and if it promotes its recruitment to the plasma membrane. And we're doing a lot of work on this, but I don't have any major new data to present on the mechanism there. So the next thing we decided to investigate was, the beauty of protein phosphorylation is that it's reversible. You can phosphorylate a site to influence biology, and then you can dephosphorylate it to switch that off. And that's the key thing about protein phosphorylation. And we were curious to know what the protein phosphatase was that acted on the phosphorylated Rab proteins. And so we did, and we thought this phosphatase was important because when you treat cells with an LRRK2 inhibitor, within 1 minute, most of the Rab protein phosphorylation is reduced, and it's very, very rapid, as you can see from from this time course here. But instead, when you look at another site in LRRK2, this is probably a casein kinase 1 phosphorylation site that becomes dephosphorylated when you inhibit LRRK2. This takes much longer time. It takes about 80 minutes to dephosphorylate this site. So this suggests that the phosphatase that dephosphorylates Rab proteins is very active and is playing an important role in keeping Rab protein phosphorylation in cells at a low level. So probably most of you will know this here, but there's around 189 catalytic subunits of phosphatases. They belong to 10 different subfamilies of phosphatase. And there's obviously many more regulatory subunits as well that control the localization and activity of each one of these phosphatases. And the interesting thing is that the vast majority of these are really unstudied. For reasons I don't really understand, is that most people seem to work in kinases and not enough on phosphatases. So there's still a lot more work to be done on understanding protein phosphatases. So what we did to see whether any of these were regulating Rab protein phosphorylation, we just did a simple siRNA screen where we knocked down each one of these phosphatases individually and tested the impact that this had on Rab protein phosphorylation. And the top hit from this screen was an enzyme called PPM1H. It's a very simple phosphatase. It's a magnesium-dependent protein phosphatase that has basically just a catalytic domain. It also has in its N-terminus an amphipathic helix that Susan Pfeffer's lab has shown is really critical for regulating membrane binding. And and as a PPM is 1H is a member of the PPM family of phosphatases that's a very well worked on in Alexander Newton's lab. And you'll all have heard Alexander talk about the FLIP1 and FLIP2, which are the major phosphatases for the AKT, PKC, and other pathways. And these are super interesting enzymes. And PPM1H belongs to a branch of this family that has 2 related homologs, J and M. So when you make recombinant PPM1H, this in vitro will rapidly dephosphorylate, the Rab proteins that can be seen in this, in this biochemical experiment where you have phosphorylated Rab8 and it becomes dephosphorylated over time when you incubate it with wild type, but not a catalytically inactive PPM1H phosphatase. This enzyme, it turns out to be completely specific for Rabs. We've given it several other phosphorylated substrates like phosphoubiquitin, even Rab8 phosphorylated at a distinct site called serine 111 that's regulated by the pink signaling pathway. And the PPM1H will not dephosphorylate other phosphorylated substrates we have presented it with. And working with our collaborator in Dublin, again, Amir Khan, we've been able to crystallize the PPM1H phosphatase. And if you look at the left part of this structure, it has a classical PPM phosphatase domain shown in green. But what was interesting, in the middle of its phosphatase domain, it has about 110 amino acid insertion that forms another globular domain that we've called the flap domain. And then we've done some mass spectrometry cross-linking where you form a complex between the phosphoRab8 and the PPM1H, and using cross-linking, we've identified a lot of the cross-links seem to form between the flap domain and the phosphoRab8, suggesting that this flap domain is a key substrate recognition domain that recognizes the phosphorylated Rab protein, positioning the threonine 72 phosphorylation site, right towards the active site of the phosphatase. And to demonstrate that this model might be correct, we've done some biochemical studies. So here, this is a biochemical study where you take, in blue is PPM1H, and it dephosphorylates Rab proteins nicely in a time-dependent manner. In red here is the most closely related relative to PPM1H. This is known as PPM1J. And so PPM1J doesn't dephosphorylate Rabs, so it's completely inactive. But if you graft in the flap domain that's found on PPM1H into PPM1J, now you go to the green chimeric phosphatase. Now this PPM1J actually becomes, if anything, even more active than the PPM1H, simply by introducing this non-catalytic domain into the phosphatase. So we think this is the key substrate binding domain, and it's going to be really important to do Chi-UM or other structure biology of the complex of the phosphatase bound to the phosphorylated Rab8. And we haven't yet been able to achieve that, but we're working towards that. And then another thing we wanted to do is to knock out PPM1H and show that this boosted phospho-Rab protein phosphorylation. So to do this, we've used a very nice new method that was developed by my colleague Alessi, who developed this tag called a bromo domain tag, bromo tag. And this actually binds this ligand called AGB1, whose structure is shown here. And this ligand doesn't bind to anything else in the cell except the bromo tag. This has been very well characterized, so it's uniquely specific. But this tag will also recruit the VH cell, E3 ligase machinery. And this will therefore induce the very rapid degradation of your protein of interest when you add the AGB1 complex. And I think this is the ideal way to study protein phosphatases, because you can knock out your protein within an hour or two using this method and study what happens to the protein phosphorylation rapidly in response to loss of protein phosphatases. So this is the result of this experiment. So we have we've epitope tagged the PPM1H with the bromo tag at the C terminus, because that's away from the amphipathic helix, and then you add in the AGB1 compound at different concentrations for 4 hours, and you can see by the time you get to 100 nanomolar and 300 nanomolar, you've completely lost virtually 98% of the PPM1H that's in the cell. And as you're knocking down the PPM1H, you're enhancing the phosphorylation of the Rab10 protein that can be also seen by the immunoblot analysis. And we've done a lot of more work on this in different time courses and doses. But you know, this gives us sort of good genetic evidence that PPM1H is indeed phosphorylating, dephosphorylating Rab proteins and counteracting LRRK2. And the final bit of evidence is that you can also make substrate trapping mutants of phosphatases. That's one advantage of phosphatases over kinases. You can carefully design substrate trapping mutants. You have to sort of test different versions of it, but we found one that worked extremely well. And then this is an unbiased way of trapping the endogenous substrate that the phosphatase works on, bound to the phosphatase. So you express the phosphate trapping mutant in cells, and then you immunoprecipitate the phosphatase, and you see what substrates are bound to it, either in mass spectrometry or immunoblot analysis. And when we do this experiment, so this D288A mutation is a substrate trapping mutant. This is a non-substrate trapping mutant, and this is the wild type. So when you pull down the wild type or the non-substrate trapping mutant, you see no binding of phosphorylated or phosphorylated. But as soon as you use the substrate trapping mutant in cells, you can see it binds to the endogenous phosphorylated proteins, which also gives us great confidence that this is indeed the physiological substrate of the phosphatase. So I'm going to finish my talk just by telling you about the homologue of LRRK2, which is called LRRK1. And so LRRK1 is quite similar to LRRK2. The key difference is that it doesn't have this armadillo domain. So it doesn't have this Rab recruiting domain that takes it to membranes. And it's not associated with Parkinson's disease. Instead, it's associated with this metabolic bone disorder that results in very severe decay of the bones, especially towards the end of the bones, in the form of osteosclerosis. And it's a extremely severe bone disorder that's caused by mutations towards the C-terminus of LRRK1. So because LRRK2 regulated Rab protein phosphorylation, we thought it was likely that LRRK1 would also phosphorylate Rab proteins. So what we did is, the first experiment we did a mass spectrometry study where we analyzed the group of phosphorylated Rab proteins in both the wild type and LRRK1 knockout cells that we generated. And these are mouse embryonic fibroblast cells. And what we found was that the Rabs that were phosphorylated by LRRK2, the 8, 10, 35, and 3, weren't affected by the LRRK1 knockout. But Rab7, which is another very well-studied Rabs, involved in late endosome biology, were phosphorylated, and they became markedly dephosphorylated in response to loss of LRRK1. And this can also be seen using a phosphospecific antibody. Here, when you blot for the phosphorylated Rab7, this is serine 72, you can see that it's no longer phosphorylated in the LRRK1 knockout cells. And when you add the LRRK2 inhibitor, this is MLI2, you don't affect Rab7 phosphorylation, because MLI2 is an LRRK2 inhibitor that doesn't inhibit LRRK1, and therefore it doesn't affect Rab7 protein phosphorylation. And LRRK1 is very selective. It doesn't phosphorylate Rab8 or Rab10. It only phosphorylates Rab7, and LRRK2 phosphorylates Rab8 and Rab10, but doesn't phosphorylate Rab7. So obviously that's a very interesting question for the toxobiologists here, is how is the selectivity for these substrates controlled? And it's even, you can see this even in vitro, that LRRK1 will very efficiently phosphorylate Rab7, but not Rab8 and 10, and vice versa. LRRK2 will not phosphorylate Rab7, and that would be very interesting to explain the mechanism that underlies this thing. And what's also interesting, as LRRK1 has very low activity in cells, we wondered whether we could boost LRRK1 activity by introducing the equivalent mutations into the sequence that are found in LRRK2 that activates the LRRK2 kinase. And what we found is that some of these mutations, for example, the one that's equivalent to the I1441G mutation, or the mutation that's equivalent to the I2020T mutation, when you introduce those to recombinant LRRK1, you significantly boost LRRK1 activity. Interestingly, the one that's equivalent to the G2019S mutation doesn't seem to boost the activity when you introduce it into LRRK1. And then the other thing we found is we wanted to see if we could stimulate LRRK1 with a physiological agonist. So we tried many different things that often stimulate protein kinases. And the thing that worked best in our hands was phosphoryl esters. And you can see in this experiment, you see a huge activation of Rabs7 phosphorylation in cells when you stimulate with phosphoryl esters that is reduced with the heterozygous mutation and blocked when you knock out LRRK1. So this is LRRK1 mediated. And this, I won't show you the data, but this is also blocked with all the PKC inhibitors that Alexander Newton's lab uses. And even there's a more recent highly selective PKC inhibitor that's been developed and that's been entered clinical trials. And this also blocks the activation of LRRK1 mediated by PMA. And we think that what we found is that PKCs phosphorylate LRRK1 at numerous sites. And in our hands, all the PKCs we've tested seem to phosphorylate LRRK1 to different extents. And then when you take the LRRK1 phosphorylated protein and incubate it with Rabs7, you see an activation of the kinase. So LRRK1 by itself has in this condition that we've used, we see no phosphorylation of Rabs7. But after phosphorylation by these different PKC isoforms, you see a 10-fold or more enhancement in LRRK1 kinase activity. And if you do the same experiment with the kinase dead LRRK1, it's not activated, obviously. So suggesting that the PKCs aren't directly phosphorylating the Rab proteins in these experiments. And we have other evidence to suggest that there's a direct phosphorylation. So the key question is, what is the site or sites in LRRK1 that's phosphorylated by PKCs? And so this has taken us a long time because we found a whole set of sites that we were quite excited about. And then we mutated them all to alanine, and the LRRK1 was still activated, suggesting that there was another site we'd missed. So we therefore use different proteases other than trypsin, just in case the trypsin was giving the very small peptide that was lost in the mass spectrometry analysis. And by doing this, and we also used this new mass spec machine that's called the Xenotoph machine that's very good for phosphopeptides, that this was probably the key as well through our ex-colleague Nick Morris. We think we've nailed the key site now, although we haven't rigorously proven it as a series of phosphorylation sites that lie within the core B domain of LRRK1. And I hope Alexandra's watching this on the Zoom call here, because we think this threonine site I think is a good PKC site. It has a phenylalanine at the +1 position, an arginine at the -3, and an arginine at the -5. And we use this conserved motifs methodology to look at the conservation of phosphorylation sites through evolution. So the highest score you can get is 9. And these key sites that I'm pointing out here are 9. There's also another site, threonine 1064. This is the one we identified originally, but when you mutate this site by itself, it doesn't affect the activation. So my money is on this 1075 site as being the key site. And this site is, I'm interested tomorrow to talk to Andres and his colleagues, because I think they've been doing some work on LRRK1 structural biology. And this site, according to at least it's an AlphaFold model, lies within the loop of the core B domain. And obviously it'd be very interesting to know how this phosphorylation might impact LRRK1 activity. So I'll just finish up now to say that we all know that Rabs are controlled by GEFs and GAPs activity, controlling the GTP binding. But what I think we found is that there's a new dimension of regulation that involves phosphorylation of the Rab proteins within their effector binding motif by LRRK1 and likely many other kinases. And then this will enable Rabs to adopt different functions, bind to different effectors in response to different biological stimuli. And indeed, there's a lot of evidence that the Rabs I show in this slide are indeed phosphorylated at their switch 2 motif, but not by LRRK2, because their phosphorylation is not controlled by, necessarily controlled by LRRK2, and probably other kinases are involved in phosphorylating these Rab proteins. And there's still lots to understand about how LRRK2 is regulated, and functions. It's clearly, its membrane localization is going to be key, and how that is regulated. I mean, obviously binding to Rabs will take it to membranes. This VPS35 complex can control LRRK2 activity. There's a lot of evidence that infection pathways and environmental toxins that cause Parkinson's disease might also stimulate LRRK2, and LRRK2 is counteracted by this phosphatase called PPM1H, and there's likely other phosphatases as well might also operate in this pathway. And over years, if you have too much Rab protein phosphorylation, this is why this biology is hard to understand, because in cancer, these changes often cause cells to grow within hours after you switch on the pathway. But for LRRK2, you might live for 50, 60 years with enhanced Rab protein phosphorylation before you get the disease. And how to study that type of biology that takes such a long time to happen, in a lab in which the students and postdocs only have months or years of work to do, is quite challenging. And I always get asked this when I give a talk about what are the mechanisms by which LRRK2 can cause Parkinson's disease. So I've told you about the work of Susan Pfeffer and ciliogenesis biology. Obviously affecting, Rabs affects vesicular trafficking, leads to lysosomal stress and defects in synaptic vesicle recycling that can obviously be linked to Parkinson's disease. In the immune system, LRRK2 is very highly expressed, and there's many very good researchers who think that when you have elevation of LRRK2 biology in immune cells, this might protect you from inflammation and infection pathways, because you boost the LRRK2 pathway. Indeed, it's possible that hundreds of years ago, a person with elevated LRRK2 activity might have a boosted immune system and be resistant to bacterial or viral infections as a result of this. And this could be quite good. I mean, no one in those days would live past their 20s or 30s to develop Parkinson's disease. But if you have too much LRRK2 in your immune system, that could lead to potentially neuroinflammation as you get older, and then this could also contribute to Parkinson's disease. And I just want to finish on where the drug therapy is with the LRRK2 pathways. There's several companies now have developed very potent LRRK2 inhibitors. Denali Therapeutics, in collaboration with Biogen, are the most advanced, I believe. They've selected one candidate, which is a compound called DNL151 that's, I think, entering phase 2, phase 3 clinical trials. They're doing trials with two groups of patients. Group 1 has LRRK2 mutations, and they want people who are just early diagnosed, because the thought is that these people will benefit most from the therapy treatment. And the second line of therapy is another group of people who are newly diagnosed idiopathic disease patients, to see whether or not they can identify a subset of these patients who might also benefit from LRRK2 inhibitors. And Biogen also have an antisense oligonucleotide approach to reduce LRRK2, and other companies are working in this area. So it will be interesting in the next 4 or 5 years to know whether this approach has any beneficial effect in slowing the progression of Parkinson's disease. And my last slide is I just want to emphasize that there's a lot of other evidence to suggest that Rab proteins are involved in Parkinson's disease beyond the work I presented in my talk. A lot of very good researchers working on the pink Parkin biology in Parkinson's disease have found that pinks can phosphorylate Rab proteins at a distinct site to LRRK2. This is the serine 111 site. And, the work of Susan, Lindqvist and shown alpha-synuclein that regulates, toxicity effect is counteracted by Rab proteins in cells. This was actually the first evidence that Rab proteins were involved. And then, mutation of another Rab protein called Rab39B that's not controlled by LRRK2 also causes a Parkinson's disease-like disorder. And, so there's a lot more work needed to understand all of this. And I just want to finish with the acknowledgements of the many people in my lab who have contributed to the work I presented that are listed here. They've all played, a fantastic role in the research. And then, we work very collaboratively. So I've been, greatly supported by my collaborator, Susan Pfeffer, at the University of Stanford for the last 4 or 5 years, and I work on this pathway. We've collaborated with Amir and his team on the structural biology aspects of the project. Sebastian and Matteo and Stefan Knapp have been very helpful in providing us recombinant LRRK1, which is instrumental in the work I talked about. And for the work on human monocytes, this was done with Alexander Zimpritz. He was actually the first author of the key paper that discovered that LRRK2 was mutated and was linked to Parkinson's disease. And he has fantastic access to patients in Austria with VPS35 mutations. And, thank you for your attention.
Can people hear me okay? I guess I'll resume. Before Dario takes any questions, we wanted to give this token of appreciation to him.
Well, thank you so much. Thank you. Thank you very much. Thank you. Oh, very nice. Wow, this is beautiful.
Yeah, this is a beautiful cute. It's a LRRK2.
LRRK2.
Alexandra got this for you.
Yeah. Recognize it. Yes, of course.
Oh.
Sorry. Yes, yeah. Oh, thank you very much. This is Alexandra. No, beautiful. Great.
And then we'll keep
this
so that you
lovely.
don't
Yes,
have to travel with you during the
This
day.
will have my pride and place. This is really fantastic. I can see all my visitors when they come.
Great.
Great. Thank you very
so
much.
Dario.
Thank you
Okay,
so much.
Now,
Yes, sure.
Dario will take questions.
Tony, yes. Do I do it with you or how? Is someone going to chair the questions or? Yeah.
So that was great. Amazing amount of new stuff. A couple of questions. Do you ever find loss of function mutations in PPM1H and
Yes, no, we were hoping that we would, because that would be clearly what one might expect.
Right.
So we've asked lots of geneticists, and there's no strong evidence that there's loss of mutations. The thing is, there's so much. Yeah, it's whether the heterozygous mutation would be enough, because the people with one copy of the gene, we've made knockout mice. So if the heterozygous mice, you don't see any effect on the Rab protein phosphorylation. In the homozygous, you do. So it's whether knocking out just one copy of the PPM1H gene would be enough to stimulate the pathway to cause. But so yeah, unfortunately we haven't seen that yet.
And the second question is about the phosphorylation of the switch 2 site. It looked like it was sort of structured. Do you think that LRRK2 sort of unfolds that region in order to phosphorylate that conserved serine threonine residue?
Yeah, that would be interesting. And so maybe Andres and other colleagues here would know better, but it's possible how that. Yeah, the peptide, we made the peptide, and that's not well phosphorylated, so the Rab protein is much better phosphorylated than just the peptide. But
yeah, I mean, there is a prior example with PKR that phosphorylates EIF2 alpha, where there is a co-crystal structure showing how the kinase unfolds the helix, the serine.
Well, that would be a nice dramatic future structure to get the structure of the Rabs bound to LRRK2 and understand that. Yes.
Hi, I loved your talk. I was curious if that VPS35 mutant enhances the membrane association of LRRK2.
Yeah, we've kind of looked for that, and we haven't necessarily seen this persuasively. It's whether this is very transient. It goes and comes off, and so with Rab29 it all goes. It's very easy to see, but with VPS35, when you look at it on the cells, you do see some differences on the where the phosphoRabs are, and we haven't looked at monocytes. We've looked at in fibroblasts and mainly in fibroblasts, I would think, but and A549 cells maybe, but we haven't seen. We haven't unfortunately seen. It's also hard to study endogenous LRRK2 localization. None of the antibodies, although people publish with them every week, you see papers. When we use these antibodies, we get the signals in the wild type of knockout cells. So this is also complicated answering this question. I think we need a better localization system. I can read them. Sorry. Yes. So what do? I click Q&A.
Okay. Roger, you can ask your question now.
Oh, thank you. I just wanted to ask you a couple of questions and follow up to Tony's question. So it was very interesting that switch 2 is so important as a substrate for phosphorylation. Do you know whether phosphorylation affects not only exchange, but also GTPase activity of any of the Rabs? Because
Yes.
it's located on switch 2, and switch 2 adopts a very unique 310 helix, which is critical for interacting with effectors. But it also, residues there also contribute to the GTPase activity of Rabs.
Yes, so we've tested this with Rab8 especially, and in our hands, and I think other groups have done this as well, the phosphorylation of the Rabs doesn't affect at all GTP binding or GTPase activity. I think everybody agrees with that. The phosphate group sort of points out, away from the catalytic moieties, so it's outward facing. So I think that's been done quite carefully, and the answer is that it doesn't seem to affect.
Yeah, I'm sort of surprised, because I would have thought that if anything, it may have decreased interaction with
Yes.
a gap, a GTPase bind accelerator.
Oh yes, I'm sure it does that. Yeah, no, I'm sure. There's no doubt that the phosphorylation of the Rab at that site will stop binding of the gap and the GEF probably.
Okay.
So the Rab will stay loaded with the nucleotide it's bound to at the time. I apologize if I didn't articulate this clearly. That's the first time I've given the talk for 2 years, so I'm a bit rusty here, but that's not a weak excuse. But it doesn't bind to the Rab. So our hypothesis is that the Rab is actually trapped in the conformation in which it's the nucleotide in which it's phosphorylated.
That makes.
It can't move as well. I should have said this. It doesn't move because we can't bind to GDI. So the Rab is literally trapped in the cellular location. It's phosphorylated, and therefore it accumulates. And you see this if you put LRRK2 onto the mitochondria. You see the mitochondria starts accumulating huge levels of phosphorylated Rab10 that you never normally see. So, yes, that's a very good point you've made. Yes, thank you.
Thank you. Yeah, I was curious. I thought there had to be some effect on GTPase activity.
Yes, no, that's a very good point.
Thank you very much. I really enjoyed your talk.
Thank you very much.
Alexandra, do you want to ask your question now?
Yeah, I did. Dario, that was a beautiful talk. Thank you very much. And
Alexandra.
sorry I went to such extreme theatrics to avoid being there in person. So I had a question about LRRK1. So PKC finds its substrates at the membrane.
Yes.
Do you have any evidence that LRRK1 is membrane localized?
Yes.
And what would control that?
Yes. Yeah, I've got slides for this, but I just took them off. But so in our hands, and here again, we have to use overexpressed LRRK1 because we don't have the technology to look at the endogenous, but the overexpressed LRRK1 is about 50% at the plasma membrane of cells. It seems to be more. And when you make membrane and cytosolic fractions, you can see this. And it's really nice when you stimulate with four-ball esters and you make the membrane and cytosolic fractions, as you've shown many times. In fact, Assad used exactly your papers. I think he was inspired by talking to you, that you can see the PKCs move directly to the membrane fraction. It's quite dramatic, actually. And LRRK1 doesn't move. So it seems that the PKCs are finding LRRK1 when they move to the membrane fraction.
And just one other addition to that. So do you know what part of LRRK1 is driving that membrane interaction?
We don't know yet. That's a very good question, you know, and yes, we don't know that question. We don't know the answer to that. That's a really good question.
Thank you.
So, Dario, so
Yes.
when different perhaps the cancer in which the kinase, when it's activated, will keep inducing signals, and if you use an inhibitor like BRAF in melanoma, it will stop signaling.
Yes.
In this case, for LRRK2, seems to be accumulating over many years.
Yes.
The question is whether the kinase activity of LRRK2 remains being essential for the Parkinson to maintain Parkinson's.
Yes.
And if it is, certainly an inhibitor would be ideal, but if it's no longer needed,
Yes.
may not be.
No,
active.
that's absolutely right. I mean, you know, the question is once you trigger the disease, whether you can stop it by by treating the trigger. And our knowledge of other neurodegenerative diseases, unfortunately, is that if you treat the trigger, you don't slow the progression of the disease down because it's already gone. But that does worry me a lot. And I guess we just have to take one step at a time. And it's important to answer the question of whether it will work or not. And I think it's 50-50 whether it would work, maybe even less than that, probably. And if it doesn't work, then you have to go and think of other downstream mechanisms that might work. And there's plan B and C, and also maybe preventative as well. So people with mutations in LRRK2, I think soon all babies being born, many will be sequenced and they'll find the mutations. And at some stage in someone's life, you would say 25, 35, is it worth taking an LRRK2 inhibitor to prevent the disease? So that might be a sort of another way of doing it. But it is a big worry whether this treatment will work or not.
Right, using the same approach for cancer, for prevention. So a patient no longer doesn't have cancer yet, but yes. if there is a chance that because mutations or SNPs or germinal mutations, they may develop cancer. So then the idea is usually to repurpose a drug that is being used safely for something else.
Yes.
To prevent. So building on that, have you done any tests, for example, I don't know, aspirin or any of these commonly used drugs that may have a subtle effect?
To see whether they would, so like metformin or something,
exactly.
that would block the LRRK2 pathway. Yeah. No, I haven't. Interestingly, the one thing that blocks Parkinson's disease, which is a bit strange, and I was the one time I met, Princess Anne, she asked me, Dario, why does smoking, prevent you from getting Parkinson's disease? So, I thought this was very strange, but it's true. People who smoke have a significantly, obviously they often die of lung cancer a lot as well. But apparently, even correcting for that. So I did have a mad idea once that you could test whether smoking could inhibit the LRRK2 pathway, and whether this could explain the results, spurred with my discussion with Princess Anne. But we're not allowed to do any research in smoking in our unit, so that hasn't been investigated.
In the interest of time, I guess 2 more questions. One from Susan, I think another from over Zoom. Oh, 4 on Zoom. Okay, 5 quick questions.
I'll answer very quickly.
Actually, Jean, we have a question from. I was going to say, we have a question from Alexander Korneff. He's had his hand up from before my time.
Yes, go ahead, ask question.
Alexander, do you want to ask your question?
That's okay.
No, I didn't mean to raise a hand, I'm sure.
Oh,
That's okay.
Okay,
Okay, now Susan.
so I'm wondering if smoking, if it has anything to do with nicotine. I mean, there's a lot of things with neuro.
Yes, it could. It could.
Yeah,
potentially.
there's number of things there. But I should know this, but what is the mechanism for the phosphatase, trapping of the substrates with the phosphatase? Is that an active site?
Yeah, no,
Yes.
it's nearby the act. So it's it's a histidine to no, it's an aspartic acid to an alanine mutation.
Mhm.
And there's I can't remember the name of the research, but they did some beautiful work. Alexandra probably does. It was a beautiful, I think, JBC paper where they tested lots of residues in PPM1A, and then they found that this mutation, obviously the phosphatase recognizes the substrate and dephosphorylates it and then dissociates.
Right.
But this mutation, it doesn't affect the recognition moiety, so you get that high affinity binding, but then it's just it doesn't. It's not able to it's not able to dissociate. I'm not sure I understand it completely, but it's a very I can send you the paper. It's a beautiful JBC paper.
So Janine, you want to call people? Greg, you can ask your question now.
Yeah, thank you. Awesome talk, Dario. So early in your talk, you alluded to the connection between, or alluded to lysosomal and mitochondrial function. And I'm wondering if you can speak to a connection with oxidative damage, and maybe that's also related to your comment about smoking.
Sorry, can you say the last bit again? I missed that. I heard the mitochondrial lysosome connection and
Connection with oxidative damage. Can you
oxidative.
speak to that?
Yes. So so there's lots of evidence that LRRK2 affects lysosomal biology, autophagy, and basal mitophagy is also controlled by LRRK2. In terms of the mechanisms, I don't think we have good connections about how this works. You know, I think the phospho-Rab effector complexes will be bind to these organelles and recruit other maybe adapter proteins or regulators. But I don't think anyone has really understood this biology. I mean, I think some of the agents that cause lysosomal damage also activate LRRK2 and induce its recruitment to the lysosome, and maybe the same is true with the mitochondria. So I think there's much more work to be done in this area.
Thank you.
Next, I don't know, Jing Yang. Before, oh, sir. Well, I guess I first wanted to say this before everybody disappears. I guess, well, first of all, great to have all of you show up despite the rain, and thanks to everybody. And I guess we had a reception planned, but I guess the exciting rainfall kind of disrupted that. But I was told there's food in the right outside, so that's one quick thing. Second quick thing is please complete the sign-in sheet for the safety reasons, safety considerations. So those are two quick reminders. And then with that, I think you can stay as long as you want. And I think Dario probably would like to answer the remaining questions. Yes. People over on the Zoom can probably just speak up because it's hard for me to
if you all want to go for lunch, you're happy to go for
you in the Q&A box.
Okay, great. Oh, you can.
I have to read them. Sorry. Yes. Does LRRK2 have any role in the heart? So the answer to that is expressed at very low levels in the heart, and I don't know of any role in the heart. So that's my answer to that one. I think we've yeah. One of the questions about the pH. How does pH regulate the activity of LRRK2 in the lysosome? And I think the answer to that is that that's a very interesting question, and we don't know the answer to that question either.
Jing Yang, Jing, can you ask
you
your
hear me now?
question?
Yes. Can you hear me now? Hello?
Yes, we can hear you. Sorry,
Oh,
yes, yeah.
okay. Yeah, it's a great talk. In terms of the phosphatase, is the phosphatase activity regulated somehow, and how do you go about it?
Yes. So how is the phosphatase activity regulated? That's a fantastic question, and the answer is that at the moment we don't know. I think it binds to membranes, and clearly binding to the membrane will bring the phosphatase in proximity with the substrate, but we don't understand how the phosphatase might be regulated, and whether there's a regulatory subunit and that controls the phosphatase activity. And I think that would be very interesting to explore.
What's the best approach to go about this? Yesterday I showed you we also have a phosphatase. I don't even know like what would you do to trying to approach this question? I guess this is trying to get your suggestion.
Okay, well, yes, thank you. No, yes, but thank you. Okay.
If feel free to contact Dario for additional questions or stay here. Yeah. Yeah, and let's thank Dario for a fantastic lecture once.
Okay, thank you so much. Thank you. Yeah, thank you so much. Well, it's nice. It's good. It's very